Next part is gene expression, that is from DNA to protein, how it happens. So in this diagram you can see the process of replication that we have already studied. Then gene expression is from DNA to protein. So this event happens uh, in two stages. First is transcription, in which DNA to mRNA synthesis is the step. Then the second step is RNA to protein. That means from mRNA to protein. That is called as translation. Now let us understand the process of transcription and translation. So this process involves the role of three types of RNAs: mRNA, that is messenger RNA; rRNA, that is ribosomal RNA; and tRNA, that is transfer RNA. mRNA is synthesized from the DNA, which is called as messenger RNA, in which the information which is present in one of the strands of DNA is transferred into in the form of nucleotides into RNA, which is called as messenger RNA. Then this messenger RNA, it binds to ribosomes. Ribosomes are the sites for the synthesis of protein. So for the binding of mRNA to ribosomes, there is there is a function, that is the function of rRNA. So rRNA, it helps in the binding of mRNA to ribosomes. And later on there comes the role of tRNA, transfer RNA. These transfer RNA, as the name indicates, are responsible for transfer of amino acids to the growing polypeptide chain. And finally, it leads to synthesis of a polypeptide chain. We call it as protein. DNA to protein. Suppose this is a cell, that is eukaryotic cell. It contains a nucleus, and this nucleus, it contains chromosomes. This is called a genome. This genome, it consists of chromosomes. These chromosomes are made up with long strands of DNA which are wound around the histone proteins. This DNA, it contains genes. These genes contains the instructions responsible for the synthesis of protein. So there is an enzyme called RNA polymerase. It binds to that gene and starts synthesis of messenger RNA. Here you can see that the one of the bottom strand of that double stranded DNA, it is so after unfolding, that bottom strand is being used as template for the synthesis of new strand. So the new strand which is being synthesized here, it is the mRNA strand which is being synthesized by RNA polymerase. So you can see here the nitrogenous bases which are being added are adenine, uracil, guanine and cytosine. So these nitrogenous bases are added for the synthesis of mRNA. So this is the synthesis of mRNA. This is transcription. In case of eukaryotic cell, this messenger RNA may contain unnecessary bases, nitrogenous bases, which are called as introns. They are cut by nucleases, And this is how there is synthesis of then this mRNA. It comes out of nucleus in cytoplasm. That happens in case of eukaryotic cell. So these are the ribosomes that binds to messenger RNA, and this messenger RNA, then there is presence of tRNA. You can see the structure of tRNA, transfer RNA. At the top, there is presence of a amino acid, and at the bottom, you can see there are sticks, color sticks, green, white, and orange. These color sticks are nothing but the nitrogenous bases. So there is presence of codons in DNA, and these are the complementary bases which are called as anticodons. So whatever the codons which are present, which are transferred into mRNA, these are the anticodons. That means these anticodons are going to bind to the specific codons. This is called as genetic code